It's been nearly 18 months since we've been living and unfortunately in some cases dying and suffering with this virus. The amazing thing is in 18 months, nobody knows where it came from or where it started and where it jumped to and from where it might have jumped to us human beings. Nobody seems to have an idea. So it just takes me back to that Amir Khan movie, Kahan se aaya tha wo? Nobody knows kahan se aaya tha. Nobody knows where did it come from. So how can this happen in 21st century with such advanced science? And what has happened as this argument and debate has gone on, something that was dismissed completely as conspiracy theories until a couple of weeks back. In fact, so much so that Facebook itself said that anybody suggesting that COVID-19 came out of a lab, whether through a leak of biowarfare, doesn't matter, it came out of a lab, would be blocked or would be banned. Now that debate has now opened up. As we know, even Joe Biden has said that he's asking his intelligence agencies to look into the origins of the virus. And for this to be open, the can is not opened yet, and we don't know what's inside the can. But somebody has really kicked this can loose. And that somebody is my guest on Off the Cuff today. We have many questions to ask him. Author, analyst, journalist, editor, and a brave person, a brave intellectual, Nicholas Wade. Welcome to Off the Cuff. And tell us, what have you started? What fire have you lit?
Well, I'm not sure if it's a fire. It's certainly a shift in public opinion. And I hope it's one that leads into putting more pressure on the Chinese government to release whatever information it may have and which it is at present holding back from us.
Yeah. So what you did was you wrote this 11 or 12,000 word article that was published on Bulletin of Atomic Scientists, which is a very respected journal and taken very seriously. The few points you made, the key points you made there were, one is this, that nobody has found an animal with this virus. Nobody has found an intermediate with this virus. And there are some features with this virus, and there's a lot of other circumstantial evidence that tells you that this needs more investigation. Now, would you have been dismissed as a nutcase if you had said this before Trump had been defeated?
Well, certainly the opinion of the mainstream media was that the virus had emerged naturally, as virologists were saying in public, and they dismissed a lab leak as a conspiracy. It was two small groups of virologists who captured the public narrative in a quite remarkable way. This should never have been allowed to happen, but it did because our mainstream media was not doing any reporting. They were too caught up in their ideological struggles. And Mr. Trump's intervention, as you mentioned, was extremely unhelpful in polarizing the issue and making people talk about politics, which they much preferred, rather than looking at the science, which editors find harder to understand.
Which side of the political divide are you on? Are you left, right?
I regard myself as an independent, as someone right in the middle.
Okay. I will not ask you who you vote for. I mean, if you wish to tell me, you can tell me that, but I will not threaten to pull out your fingernails. This is not China. So we can't do those things. Now, what is it? First of all, can you summarize what you said? The gist of your 12,000 words.
Yes, I tried to lay out a way of looking at this issue that was a purely scientific way that stepped clear of all the politics. And I said, it seems to me very obvious that there were two hypotheses on the table. Both were very plausible and reasonable. One was that the virus had jumped from an animal to human, as many epidemics have before it. And the other was that the virus had escaped from a lab. And right there in the middle of Wuhan is the leading lab that does research on coronavirus in China. So obviously, since we know that viruses do escape from labs quite frequently, there's certainly a possibility that a virus had escaped from this lab. So you have two plausible hypotheses on the table. There's no direct evidence for either one. It's important to keep that in mind. So we have no final proof or conclusion. But if you take these two hypotheses and you take the available evidence and say, well, which of these two hypotheses explains the evidence better? So that's what most of my article is concerned about, just weighing up the evidence and seeing which hypothesis does better. And in every case, I find that lab leak provides a better, more plausible explanation than does natural emergence.
So you are leaning towards lab leak, but let me also clarify, lab leak does not mean biological warfare.
Absolutely. There's no evidence for biological warfare. And we know that virologists all around the world have been doing this kind of experiment. Their rationale is that if they take a dangerous animal virus that might spread to humans, and if they can study it in the lab and alter it in ways that would make it more infectious for humans safely inside their lab, then they can get a jump ahead of nature and be able to predict and prevent the next epidemic. That's the rationale. Many people may not agree with it, especially in the light of recent events. But the Chinese biologists in this regard, so far as we know, we're not doing anything very different from what virologists around the world have been doing. It's just they were working with a particularly dangerous virus, the coronavirus.
And also particularly in a system which is quite close. It's not like Germany or US or India or Britain. It's China, which is quite closed.
Yes, the Chinese system is closed. Of course, its scientists seek out and have collaborations with scientists in other countries, but everything is basically under the control of the Communist Party.
Yeah. So you said you also added one more expression to our vocabulary, that is gain of function. I know that this is something that a very small community of virologists, bacteriologists, microbiologists might have known, but we didn't know anything about it. So gain of function, if I try to sort of oversimplify it, as I often do, because in a way, I'm a science graduate from a long time back, but I'm a failed science graduate in the sense that I did no science after that. So when I try to oversimplify it, I say it's like lots of people are lined up before a race begins and somebody takes a start ahead. So that is taking a head start ahead of the others, you break some rules. But when you say that I am running, but I will give myself some prohibited substance that Olympic rules will not allow, that will help me get ahead of the race so I can look back and see who's running how. That is a bit like gain of function. Am I making it more complicated or am I oversimplifying it?
Well, I think gain of function is something more than just getting ahead in the race, though, of course, virologists, like other scientists, are very competitive. Gain of function simply means when you take a natural virus and you alter its genome to give it qualities it did not have before and which make it more infective, more able to become a pathogen. So gain of function is a very deceptive phrase because it sounds so mild, but actually it involves creating a dangerous virus that did not exist in the world before. So there's been considerable debate about this issue more or less for the last 20 years, ever since virologists gained the genetic tools to alter viruses. And of course, it's a very sort of tempting technology for them. It gives them a sense of power to create something that didn't exist before in nature. It helps their careers. So there are all kinds of reasons why virologists were interested in monkeying around with these viruses, but there is the possibility there of creating something extremely dangerous.
I think scientists of all kinds and strategists love to create these cynical euphemisms. So to me, one of the comparable things here is collateral damage, isn't it? or what was it? Enhanced interrogation methods.
It certainly is a euphemism. You're quite right. It's a very sort of bland, mild phrase that makes you think, well, there's nothing much going on, but there is a lot going on. And there's been a sort of fierce debate in the United States and elsewhere in the last 10, 15 years as to what experiments should be allowed by gain of function and what experiments should be prohibited. And there was a moratorium for a short time from 2014 to 2017 on doing this kind of research, but it was subsequently lifted. So at present, the restrictions are not as severe as many people would like to see.
You saw the congressional hearings where Dr. Fauci was asked these questions. I bet you watched it very carefully. What did you make of that? You thought Dr. Fauci honestly knew only this much and had no reason to be suspicious. And now he's saying in some of the interviews that he can't be so sure. So where do we place him in this debate now?
Well, I was very puzzled when Dr. Fauci first said that. And to immediately answer your question, I think the answer is we don't know yet. It's still, there are many facts that still have to come out. I thought that because of this moratorium in place and because the National Institutes of Health, Dr. Fauci's agency, was continuing to fund this research at the Wuhan Institute of Technology, that they must have invoked a loophole in the moratorium. The moratorium allows for several exceptions to be made in special circumstances. So I assume
national security, public health, many of those.
Yes, exactly. Either of those, extreme public health or national security. So I assume Dr. Fauci had invoked one of these loopholes in order to allow the research to go ahead. So I was very surprised when he said, no, he hadn't allowed any research to go ahead. And if you look back at the language of the moratorium, it's very, very loose and allows you to do lots of research that others might think was gain of function, but the language of the moratorium says is not gain of function and is expressly allowed. So I think this is what Dr. Fauci was referring to. He was referring to the fact that the moratorium does not apply to wide categories of research in which viruses are manipulated, and therefore he had supported none at the Wuhan Institute of Virology. However, it's clear that the Wuhan Institute of Virology, particularly Dr. Zhengli Shi, was doing, they published an experiment in 2017, which seems to me to go way beyond the terms of the moratorium, however loose its language is. And I think we're going to see in the next few weeks Dr. Fauci being asked, well, even if he didn't think he was supporting any gain of function research at the Wuhan Institute, is this research published by Dr. Xing a violation of the terms of the NIH grant to Wuhan? And I think at that point we may see Dr. Fauci say, well, Dr. Shi was doing illegitimate research. She was doing research which I did not authorize, but was forbidden by the terms of the grant we gave her. Now, I don't know that's the case, but I think it's possible. This is how the situation will turn out.
Now, you mentioned this research that came out in 2017. Will you tell us more about it? Because to me, this sounds new now.
Well, I think the moratorium reflects this sort of fierce debate going on among scientists in the US and elsewhere as to how much genetic alteration of viruses should be allowed to go ahead and whether the benefits of doing so outweigh the risks. So, it's like men struggling under a tent. You can't see exactly what's happened, but I think when the moratorium was first imposed in 2014, that was a victory for the group of scientists who are against changing viruses. But I think then Dr. Dr. Fauci, who has been on the side, I think, of doing more research rather than less. And that's okay. It's probably his job. He is director of the largest research agency supporting virology. Dr. Fauci then sort of won the battle because he managed to shape the language of the moratorium in such a way that it allowed almost any research to go ahead. So the moratorium was then replaced. It came to an end in 2017. That was a victory for the sort of pro-research scientists, and it was placed by a very sort of weak reporting system that said if you're doing something that you think is really, really dangerous, you must report it to the US government. So, of course, not many virologists are going to report their experiments to the government. So this reporting system has been very, very weak. So that's what I think we see happening, the sort of to and fro as this battle takes place in the corners of in the corridors of the bureaucracy in Washington.
Yeah. So you mentioned this particular research that was published in 2017 by Shi Zhengli. What exactly was this?
Well, she was investigating the properties of some bat coronaviruses which she had found and which she had reason to believe might be very infective and cause the next jump to humans. Remember, the background of all this is that we have already had two serious epidemics from these bat coronaviruses, the SARS-1 in 2002 and the MERS in 2012. So all these coronaviruses were a matter of legitimate research of interest both to Dr. Shi and to Dr. Fauci. So Dr. Shi was taking some of these new coronaviruses, and she was studying their spike proteins. So the spike proteins are the things that jut out all around the surface of the virus, and they are the things that latch onto the target cells. And the precise nature of the spike proteins is very important because that determines what kind of host the virus is going to attack, whether it's going to be a bat or a human or a cat or whatever. So she was cutting out the genes for these spike proteins, and she was swapping them into the genome of another virus. So she could study the property of the spike protein in a sort of standard background. So she had these, I think it was 8 coronaviruses, and she was putting their spike proteins into the backbone of another virus in order to see if she could create a more powerful and effective virus. And two of these viruses, I'm sorry?
A super virus?
Two of these viruses were indeed more infective. They infected human cells, the cultures of human cells. So these were definitely gain of function. She had taken something that did not exist in nature, and she created a combined virus. We call it a chimera. She created a chimeric virus that could infect human cells. So this was way beyond the terms of the moratorium, in my view, and the view of several scientists I've spoken with.
So would you believe Dr. Fauci if he says tomorrow he had no idea this was going on?
Well, I think he's an honorable public servant, and my instinct is always to believe what he says, but then I have to try and reconcile what he says with the available facts. So if he says he didn't know what was going on, yes, that would be perfectly possible, because he's not directly overseeing this grant. He's overseeing hundreds or thousands of grants. This particular a lump of money, it didn't go directly from Dr. Fauci to Dr. Shi. It went to a man called Dr. Daszak in New York, who runs something called the Eco Health Alliance. And he, in turn, then funded Dr. Shi. So Dr. Fauci is sort of some way removed from this particular experiment. And I would think that it's probably up to Dr. Daszak, first of all, as the principal investigator, they call it, the person who's responsible for these funds. It's up to Dr. Daszak to see that these funds are being used correctly. So, if Dr. Fauci said he didn't know about it, I would tend to believe him, and then to ask why Dr. Daszak didn't know about it.
And Dr. Daszak was among the first to jump to the defense of the Chinese, to say any discussion on whether this is not a zoonotic virus is baloney. Don't do it. He wrote a letter first and did not disclose his conflict of interest.
Exactly, sir. He wrote this letter was very important. It was one of two letters published by virologists that sort of shaped public opinion for the next year, and they should never have been allowed to get away with these letters without the press examining them and probing into exactly what they meant and finding out all the sort of deep flaws in these letters. So Dr. Dashak was the author of the first. It later turned out he drafted the whole letter and got several people who were his friends to sort of sign on. And they published this letter saying, let's stand shoulder to shoulder with our fellow Chinese scientists on the forefront of lots of stuff like that. And also saying lab escape is a ridiculous conspiracy theory and we shouldn't hear a word of it. So, as you say, he had the acute conflict of interest, which he not only did he not declare it, the letter concludes by saying, we declare no conflict of interest.
Fantastic. And then there was a second letter. And all of this is, just for our viewers, all of this is happening in February of 2020, when the virus is just coming out in the world, when the world is, and a lot of people think, even us in India, we thought that this will remain in some countries like SARS and MERS, and this will not go to all of the world. And WHO is still figuring out what this is. At that point, these two letters have got published, isn't it?
Yes, that's exactly correct. The Lancet letter was in February of 2020, and the second letter of a group headed by Christian Andersen, a virologist at Scripps, that came out in Nature Medicine in April 2020. And these captured the narrative that all reporters believed. No one sort of looked into exactly what the letters were saying or the arguments they were putting forward, all of which were filled with holes when you looked at them closely.
And the second letter among its signatories had a prominent virologist called Ralph Baric, who is now also signed a letter saying why we should look at the origins of the virus.
Are you referring to the new letter in Science, May 14?
Yeah.
You must be referring to Dr. Ralph Baric.
Yes, Ralph Baric,
the leading coronavirologist in the US.
Ralph Baric, I think from North Carolina University.
That's right, yes. Now, I can't remember if he was a signature of the Andersen letter. I don't think he was, but in any case.
It's signatory of this letter.
Right. Yes, it's very significant he signed this letter because he's a leading authority on these viruses. He trained Dr. Shi himself in how to swap these spike virus genes around from one virus genome to another. So he's certainly a leading expert, and I think it is very significant that he signed the May 14 letter a few days ago.
Yeah. So what kind of pushback have you got since you wrote this?
Well, to my surprise, I haven't gotten any pushback at all. I thought it would be a controversial article and lots of people would attack it, but no one has. I think it's because I did my best effort to lay out the evidence as carefully and as unambiguously as I could. And so far, everyone has accepted the outline of the argument I presented, and I haven't seen so far any pushback.
But you have your share of critics, particularly the book you wrote on genetics, race. A lot of the scientists get together to question it.
Well, that was an earlier book I read that was controversial. It was attacked by a small group of scientists in a sort of long letter with lots of signatures. So whenever you see lots of scientists signing on to something, you should always smell a rat, because after all, science works.
A rat or a bat?
A rat in this case.
Yeah.
So science works on facts. Politics works on consensus. So if you've got the facts, you only need one person to say, well, here are the facts. If you don't have the facts, then you need to gather up a lot of scientists to sort of make it seem the weight of scientific authority is on your side. So this is how the critics attacked my last book, which was about the biological nature of race. I think it was a very good book, even though I'm the author of it. There is not a single scientific error in it that is, anyone has pointed out. There's not a single racist statement in it. These scientists were attacking it for political reasons, in that there's American universities, I hope this is not true of Indian universities, American universities have been overtaken by an extremes of delusional political left creed in which obvious facts are denied. So they deny that there's any biological basis to race. They deny that there's any difference between the sexes. They say everything is socially constructed. And anyone who disagrees with them, they will attack and harass and intimidate and try to have them thrown out of academic life. So of course, if I'd been an academic, my career would have been ended by this attack. And that's why no academic ventures into this very controversial territory. But since I'm not, I didn't care what they said. And in any case, none of this has anything to do with the origin of SARS-CoV-2.
Yeah. But if in one line you can explain to us, because now we are all learning about you, explain to us what is it that you meant in that book by way of biological origin of. Just explain it to us.
Shekhar, your sound cut out. Could you repeat the question? What does I mean by?
Give me in a line, what was the conclusion in your book that triggered so many scientists and that triggered this entire academia?
Well, it wasn't anything directly in my book. It was simply that the book, showed why there is a biological basis to race. It pointed out, if you take any animal population, you spread it across the globe, then you're going to get regional variations in each place because you don't have proper mixing. If you mix up the gene pool thoroughly every generation, everyone's going to be exactly the same. If you let nature take its course, then people will gradually evolve into sort of local variations. It's just a biological process which everyone accepts for every other species. It's just as soon as you start applying Darwinian principles to humans, ever since Darwin first wrote Origin of the Species, his theory has a great capacity to upset people in many different kinds of way.
Yeah. So back to this situation, I have some questions now. So Bhaskar Karkera, who's a banker from AU Bank, he says, how is this virus so strong that it's giving multiple waves and is changing character so quickly? Are these viruses usually like that, or is it because it was probably designed at source to multiply like this? It is to that extent a lab-made super virus.
Well, it's a good question, but I'm not expert enough to know the answer. It certainly was being trained up, if you like, in humanized mice. That's mice that are genetically engineered to have human cells in their airways. So this may have been one factor in its potency. Certainly as soon as it emerged, it was very well adapted to human cells. Other factors may simply be the nature of coronaviruses and the fact that our populations have not been exposed to them. This is a novel virus to which the human population has no natural immunity. So I think all these factors come together, plus others that I don't know about or which have not yet been discovered.
Yeah. So Anirudh Datta, who's a banker, who says that between November 2020 and now, I know we discussed some of this, what was the one thing, besides your article, of course, what was the one thing that changed the mood on this, that until now, anybody who questioned the zoonotic origins was poo-pooed, being laughed at. So what is the one other thing that changed it?
Well, I think the stage was set by the visit of the World Health Organization inquiry team to Beijing in February of this year. So at first, this seemed to be a propaganda victory for the Chinese. After all, they'd more or less handpicked the members of the committee as a condition of letting the WHO into Beijing. The WHO had to agree to all kinds of conditions of what they would not see, would not ask to see. And of course, they came out with the pre-scripted conclusion, lab escape is extremely unlikely. We won't waste any time on that. And we think it was natural emergence. So at first, this seemed like the Chinese had prevailed again in their version of events. What also became clear at that point was that the Chinese authorities had absolutely no evidence to offer the group in favor of natural emergence. So all these previous epidemics have left copious traces in the environment of their passage. You find the bat population the virus came from. You can find the intermediate animals. These were things called civets in the case of SARS-1 that are eaten in Chinese wet markets. You can see the virus mutating a few times as it gains the capacity to infect civets, which are rather different from bats. So the virus has to make these mutational shifts. It shifts a few times more when it moves to humans and becomes a sort of mild pathogen. And then after a total of about, I think it's 25 mutations, you can see SARS-1 becoming a really fierce human pathogen. So with SARS-2, at first everyone expected to see similar signs in the natural environment, the bat population, the intermediate hosts, the infected human populations. But there's none of that. There's not a shred of evidence of natural emergence.
We gave a bad name to many animals. We said it's pangolin. We said it's snakes. All kinds of animals were given a bad name.
Yes, the pangolins, I think, was a diversion carefully prepared by the Chinese. Everyone was looking for an intermediate animal. So the Chinese said, well, here's some pangolins who've been infected. But there's something wrong with the data in that report. So I think we should forget about pangolins. And once you rule the pangolins out of the picture, there's nothing there. So far, we haven't found any piece of evidence. So this set the stage, I think, for people to be a little more open to alternative hypotheses. It was 15 months since the epidemic arrived, and with no evidence, one just had to sort of be more open-minded to what might happen. That was, I think, the primary event. And then my article came out, which seems to have been quite widely read, and people sort of realized, well, yes, lab escape is certainly a very reasonable possibility. And then we had the letter from the scientists, the May 14 Science, saying, well, we should look into lab leaks seriously. And almost overnight, public opinion seems to have changed.
Because as you said in your article, with SARS, the first SARS, they found within 3 or 4 months that civet cat was the intermediate. Civet, which is not a cat, but called a cat, one of those ironies. And in the case of MERS, it took them a bit longer, maybe 8 or 9 months, but they figured that it jumped to a camel, and then from camels to humans. But in this case, nearly 15 months now, there is no sign. Now, question from Dr. Arti Parulekar, who praises your article, says it's a whistleblowing article. And she says, after this, do you think the world should form an organization to monitor strictly gain-of-function research, similar to nuclear testing regulations under, say, Comprehensive Test Ban Treaty and stuff like that?
Well, I think that would be perhaps be overkill. International organizations are often not as successful as we would like. But I think we should go back to each country having its own scientists take a hard look at gain of function and making their own rules, being satisfied that their rules are strict enough.
Sudipta Roy, who's another banker, who's quoting this report that just came out. I think I saw it in Daily Mail, so I was not that impressed to begin with. But she says that British doctor Angus Dalglish, if I pronounce it right, and Norwegian virologist Birger Sorensen's claim that 4 amino acids in a row on the COVID spike protein cannot occur naturally and must have been engineered in a lab.
Well, this was a paper that came out 3 or 4 days ago. It's quite interesting, but I'm not an expert on this subject. My feeling was that they haven't done sufficient work to show that these segments of the spike protein, which they say are unusual, in fact do not occur in other coronaviruses. So they're making a rather bold claim, saying there are these 5 or 6 regions where the spike protein has unusual sequences, which we think were sort of inserted there in a lab. And if this were the case, I just feel that other scientists would have spotted these sequences much earlier. There is one sequence in the spike protein called the furin cleavage site, which is very unusual, and there's good reason to think that could have been inserted in the lab. But I haven't seen anyone else talk about these other sections they say are novel.
In fact, that is the furin cleavage that we talk about between protein S1 and S2. And as I try to describe it to my readers and viewers, is that it's a bit like a guided missile. There is a sensor ahead that directs a missile to the target, whether it's seeking heat from an engine or a radar waves or whatever. And then as it gets the missile to the target, it sort of drops off, and then the warlord takes over. Then the explosive takes over. And as you explained, furin is like an enzyme that our cell gifts this virus to cut it as the S1, S2 cleavage. And that is to you funny.
Yes, it's very curious because the little section of protein at the furin cleavage site is sort of long enough that it's the kind of thing that viruses often obtain from one another by swapping genetic material. And they often do this when 2 viruses happen to infect the same cell. And when their genomes are assembled inside the cell, they get reassembled with bits of the other virus. So viruses can acquire new things like a furin cleavage site by borrowing them from another virus in the same family. So what's significant here is that there's no other virus in the SARS-CoV-2's family. They're called sarbecoviruses. There's no other sarbecovirus that has the furin cleavage site. So that means SARS-CoV-2 could not have acquired this extra section in the usual way by borrowing it from one of its family members. So you then have to ask, well, how on earth did it get there? And the geneticists can sort of make up various explanations, but they're very ad hoc. The favorite one is, well, SARS-CoV-2 didn't originally have a furin cleavage site because he couldn't borrow it from any other sarbecovirus, but after it jumped to humans, human cells have this sequence, so the virus could have captured it from a human cell. Well, maybe, but that's just sort of hand waving. No one has found this human population in which this process of SARS-2 capturing a furin cleavage site could have happened. And meanwhile, on the lab escape theory, there's a very easy explanation, which is simply that virologists have known for years the way to soup up a virus is to insert a furin cleavage site.
Yeah. So Occam's razor, that's why shifts to lab leak.
Yes, I think so. That's one of the pieces of evidence that lab leak can explain easily and natural emergence cannot. So the more such pieces of evidence you have, the more likely lab leak is to be the explanation.
Now, you took this evidence, you talked to Professor David Baltimore, and that name jumped up at me because he had come visiting India many years ago, 2008, I think, and I had a TV show those days called Walk the Talk, and I had featured him on that show on the grounds of the National Institute of Immunology in India. I know that he's a 1975 Nobel laureate, so he's been around, but he's a he's at Caltech, he's all there. He said that for him, this furin cleavage looked like a smoking gun.
Yes, he used that phrase, and then he went on to say this is a very serious challenge for natural emergence. So they definitely caught his eye immediately. And obviously that's not proof, but that's just his personal reaction. He is, as you say, one of the world's leading virologists, and no one will take his words lightly.
Yeah. So Rajesh Rao from Nokia, this is precisely, he said, he used the expression smoking gun. So that came from David Baltimore. Rohit Vaidya from University of Illinois in Urbana-Champaign. Unfortunately, I have to read this out. I can't have all of them present. He says, apart from accountability and assigning blame, what more will we gain by scientifically investigating where this virus came from? Did it come from a lab or not? Or did it come from a gain of function experiment or not?
Well, the most important thing we will gain is that if it did come from gain of function, that will give us very important information about how strictly we should control these experiments to make sure that nothing like this ever happens again. I think it's very important to know where the virus came from.
Karan Johar of Shloka Events, it's my question also. How has WHO come out in this as yet? I mean, has it come out with its prestige intact, or some of it intact, or respect?
Well, I'm afraid it's come out with its reputation severely damaged. It's made clear that it's in the pocket of the Chinese. And China's always had considerable control over the WHO. For example, it's managed to exclude Taiwan, which has just as much right to the World Health Organization's benefits as anyone else. So, and I think China overplayed its hand in this case because it showed how completely the WHO was in its pocket. It sort of handpicked the members of the committee. It sort of led the committee around by the nose, saying you can't go here, you can't go there. So I think the WHO has suffered a bad blow to its reputation. It should never have accepted the invitation, I think, on the Chinese terms. It needs now to reestablish its independence by showing the world that it's a true scientific body. It's independent of any government, including especially that of China, and it needs to turn over a new leaf.
If you've seen that congressional hearing, you recall Senator Kennedy's expression, if you hold Xi Jinping upside down and shook him, WHO will fall out of his pocket. So as you mentioned this, I was remembering that. Yeah. Deepa reminds us of a movie, I'm Legend. And she says in that movie, humanity was destroyed by a supposed cure for a virus. Do you see any parallel in what's happening now with the coronavirus and its vaccines?
I haven't seen the movie, so I can't really comment.
Okay, but do you think that this quest for the final cure for the final virus, this could lead us into more dangerous places?
Well, yes, I think there's a lot of danger here. I mean, nature's not necessarily our friend, and if we mess around with nature trying to create more dangerous viruses, we're going to succeed. So this is a very serious lesson. If indeed the virus did escape from the lab, we need to rethink how we do these things.
Now, there are some people who want also have views on what will happen to the rest of us. So there is Payal Sahaya who wants to know your thoughts on the topic of herd immunity. Do we have any clarity yet on what might be herd immunity from this virus?
I'm not an expert on immunology, so it's better for me to say nothing, I think.
But your own, this is my question, your own confidence in vaccines now.
Well, the vaccine seems to be amazingly successful. I mean, we were so lucky that this new type of vaccine, the RNA vaccines, came along so quickly. It was available just at the minute we needed it, and it's made an enormous difference. So everyone should be vaccinated as soon as they possibly can.
I've taken more time than I threatened to take from you. Nevertheless, just a couple of last questions. Harry Kulkarni, does science shape science fiction, or is it the other way around? I know that you don't write science fiction.
I'm sorry, is what science fiction?
Does science shape science fiction, or is it the other way around?
Well, I think usually science shapes science fiction, but there are examples of science fiction writers being ahead of science. I think the most famous one is the science writer Arthur C. Clarke, predicted communication satellites back in 1940. So I think quite often the science writers do tell us where science is going to go.
Yeah. So I have the last two questions from my colleagues. One from Arani Basu, our subscriber. And this is a question that you answer in your article, where you give possibilities on your article. But how could a virus like this escape from a high security lab like this, that too in China?
Well, there's a simple answer to that, which is that although there is a high security lab in China, it's called BSL Level 4, built for them by the French for some reason, none of Dr. Xi's experiments took place in this lab.
I see.
She's written herself that they took place in the much lower levels of BSL-2, BSL-3. BSL-2 is nothing much, but about the same safety standards as a dentist's office. So it's very easy for a virus to escape from a lab like that.
So just for our viewers, BSL stands for biosafety level. Is that correct?
That's correct. Yeah.
Yeah. So the more dangerous things you are dealing with, the higher the biosafety level. So just as an explanation, the Indian company Bharat Biotech, which is making the dead killed coronavirus vaccine, has BSL-3 facility where this is done, because they first get live viruses. So just to explain to you. Now, two last questions from my colleagues from my science bureau. Mona Basu, do you think that speculations of lab leak will now be a step back for public trust in science in the US, which already saw a significant pushback to vaccination and masking policies?
Well, that is a very interesting question, and we don't know how this is going to play out. I think if it turns out this terrible pandemic was caused by a scientific experiment, it certainly will be a blow for virology, certainly, maybe for biology as a whole. I don't know. We just have to see how it plays out. And of course, the other element of the question, there has been lots of controversy about, who should wear masks and when and wherefore. And this too, I think, is a sort of burden that scientists have to bear. But that's the nature of science. Science produces conflicting information, and it's hard for scientists to figure it all out. And so I think these sort of public debates among scientists have perhaps unavoidably helped impair its image in the public eye.
Our science editor, Sandhya Ramesh, who should have been doing this conversation, not I, but you are such a star that I grabbed it for myself. He says your article uses the furin cleavage site as an argument that claims that all of beta viruses, only SARS-2 has it. But virologists have refuted this, saying that other beta coronaviruses, including HKU1 and MERS, have it as well. And experts have explained that template switching is the likely mechanism for its development. How does it affect your argument?
Well, I think they've misunderstood what I've written. It's true that other coronaviruses do have the furin cleavage site. But what is important here is that the particular family of coronaviruses to which SARS-CoV-2 belongs, the SARS-Beco viruses, none of them that we know so far, except for SARS-CoV-2, has the furin cleavage site. That's why it's so unusual in SARS-CoV-2.
So which category of viruses you said? Sorry, I'm just idiot-proof it for me, please.
They're called SARS-Beco viruses. I tried not to use the technical term in my article, and maybe I should have done. I said instead SARS-2 related viruses, hoping everyone, the scientists at least, would understand. Well, when I said SARS.
We'll Google and spell it on the screen.
Okay.
Unless you tell me, how do you spell it?
You spell it SARBECOVIRUS.
What? Okay. SARS-CoV virus. And
SARS-CoV-2.
yeah. And the last question again from Sandhya, our science editor. How's mainstream media treating the lab accident story? Why did you have to self-publish your humongous piece first instead of writing it for a publication? New Yorker, New Yorker, Atlantic.
That's a very good question, but I think I would rather not discuss the publication history of the article.
maybe we leave it for the next one, or when you write your memoir.
All right, good idea.
But thank you so much, Nicholas Wade. You made a big, huge contribution. And I said, you haven't opened this can of viruses yet, but you've kicked it hard enough for it to be loosened. And enough people are now asking questions. And I think at the end of the day, humankind will benefit from this. So thank you very much. And thank you for your time and your patience.
Thank you, Shekhar. It was a pleasure to chat with you.
Thank you, Nicholas. Bye-bye.
Bye-bye.